Hello friends, this is the fourth class of genetics, and before I really start with the fourth class, you should watch my videos from genetics part 1 to part 3. And this is my website easyscience.pibli.com, where you can find some more videos, and this is my YouTube link where you can find videos. You can leave your comment and you can ask me some questions. I will try to answer them, and please subscribe my channel. So now I would like to start the part 4, where I will be discussing on about the duplicate recessive interaction. And let me write it. Okay, so duplicate recessive interaction, and here the ratio that we get is 13:3. Now sometimes this kind of duplicate recessive interaction is also called dominant suppression. Now what happens, that suppose if we have a white substance here, this is the white substance, let me put it in a box, and this A gene is encoding a specific enzyme and which gives a color, whereas this B gene inhibits this reaction. And suppose if this B is dominant over A, then this B which is dominant will prevent to produce colors, and most of the flowers will be white in color due to this B gene, and some of those flowers which will have a genotype carrying this small b, they will only produce the color. And along with that, this small b, this one of the functional allele has to remain present. So in this case, this capital A is the functional allele and this small a is the alternative form of the gene A. And when this gene A, which is responsible for this color product to be produced, has to be present in minimum one functional number, and so this one will produce two colors. This one will produce the colored phenotype, and this one will also produce the colored phenotype, but rest of the genotype will not be able to produce any kind of color, so it will be a white phenotype. So it is very much similar to the dominant epistasis, that is, this dominant allele of one gene suppress the expression of the another gene. Alright, so now if we go to the this Punnett square, then we can see that in these places where this capital A is present, this 3, this is this genotype carrying this capital A small a and capital B small a, capital A small a and small b, two such kind of genotype are present and one such kind of genotype is present. So overall these 3 will produce color. This 3 will produce color, and the rest of the genotype, that is this 13 genotype, will be white. That means all this 13 genotype will produce white color. And here in this one, look at this, that small b is present, but still there is no color. Why? Because the capital A is also not present here. A capital A, which is the functional allele required for the color production. Here again this 2 recessive format of this A allele is present. So in that case this will also come under the expression, the white expression. So now if again let me go back to the previous slide, and we will try to discuss another kind of another kind of epistatic interaction, which we call the duplicate dominant interaction. That is duplicate dominant interaction, and here the ratio is 15:1. So here what happens, that suppose this is a white substance, this is the white substance present in the flower. All these experiments had been done on the flower colors, so that's why I'm talking about all these things, that white substance was present as the basic color of the flower. And when this capital A is present, this capital A gene which is encoding an enzyme, and that enzyme is working on this substance and produces a kind of a product that will be colored. This product will be colored, and when again this B gene is working, here also the product will be colored. Alright, so in both the cases where if either of A or B is present, it will form the colored product. If none is present, the product is white. So in that case, this small a, small b, this is the only genotype that will produce the white color, and the rest of the genotype will produce the color. That's why this 15:1 ratio we can see. And now if we go to the Punnett square, then here we can see that this last one, this small a, this is the only genotype that will produce white color. But look at the rest of the genotype, or every genotype is containing either A or B. Alright, so this is the duplicate dominant interaction. Now again go back to our previous slide, and let me talk about another kind of epistatic interaction, that is this dominant recessive epistasis. That is called this dominant recessive epistasis. And here the kind of ratio that we observe is 9:7. How this kind of ratio we get, that one of the dominant allele, here also more than one gene is interacting. Suppose this is gene 1 and this is gene 2, and it has got two alternative form, and here it is there are two alternative form. Now one dominant allele from both the interacting gene is necessary. That means this capital A and capital B is necessary for the dominant phenotype, which is 9, to take place. That means this A, 1A, minimum 1A and 1 capital B has to be present in the genotype for this dominant expression of 9, and the rest of the genotype will produce this recessive expression, that is 7. Now if we go to the Punnett square, there we can see that this okay, so how many are there? that 1 2 3 4 5 6 7 8, and there has to be another one which will be actually okay, let me first clean this one up a bit so the thing will be easy for us to handle. Anyways, this where is capital A, small b, both, all these things are present. There are 4, there are 6 and 3 9. Okay, so 9 is already done. So now all these 9 will 9 are present, where both these capital A and B are present, that is 9, and the rest of this genotype denoting it in a different. Okay, so all these rounded genotypes 1 2 3 4 5 6 7, so where either capital A is not present, that means the phenotype might be either carrying this small a, or this genotype is carrying this small b, and all this will give different 7 kinds of genotype, which will give the recessive interaction, which will express the recessive phenotype of this dominant recessive epistatic interaction. I hope that this epistasis videos were helpful to you. I'll see you in the next video. So take care.